Section 13.3 Protists. By the end of this section, you will be able to describe the main characteristics of protists, describe important pathogenic species of protists, and describe the roles of protists as food sources and as decomposers. Figure 13.3 shows a variety of protists. Eukaryotic organisms that did not fit the criteria for the kingdoms Animalia, fungi, or plantae historically were called protists and were classified into the kingdom Protista. Protists include the single-celled eukaryotes living in pond water, although protist species live in a variety of other aquatic and terrestrial environments and occupy many different niches. Not all protists are microscopic and single-celled. There exist some very large multicellular species, such as the kelps. During the past 2 decades, the field of molecular genetics has demonstrated that some protists are more related to animals, plants, or fungi than they are to other protists. For this reason, protist lineages originally classified into the kingdom Protista have been reassigned into new kingdoms or other existing kingdoms. The evolutionary lineages of the protists continue to be examined and debated. In the meantime, the term protist is still used informally to describe the tremendously diverse group of eukaryotes. As a collective group, protists display an astounding diversity of morphologies, physiologies, and ecologies. Characteristics of protists. There are over 100,000 described living species of protists, and it is unclear how many undescribed species may exist. Since many protists live in symbiotic relationships with other organisms, and these relationships are often species-specific, there is a huge potential for undescribed protist diversity that matches the diversity of the hosts. As the catch-all term for eukaryotic organisms that are not animals, plants, fungi, or any single phylogenetically related group, it is not surprising that few characteristics are common to all protists. Nearly all protists exist in some type of aquatic environment, including freshwater and marine environments, damp soil, and even snow. Several protist species are parasites that infect animals or plants. A parasite is an organism that lives on or in another organism and feeds on it, often without killing it. A few protist species live on dead organisms or their wastes and contribute to their decay. Protist structure. The cells of protists are among the most elaborate of all cells. Most protists are microscopic and unicellular, but some true multicellular forms exist. A few protists live as colonies that behave in some ways as a group of free-living cells and in other ways as a multicellular organism. Still other protists are composed of enormous, multinucleate single cells that look like amorphous blobs of slime, or in other cases, like ferns. In fact, many protist cells are multinucleated. In some species, the nuclei are different sizes and have distinct roles in protist cell function. Single protist cells range in size from less than a micrometer to the 3-meter lengths of the multinucleate cells of the seaweed Caulerpa. Protist cells may be enveloped by animal-like membranes or plant-like cell walls. Others are encased in glassy silica-based shells or wound with pellicles of interlocking protein strips. The pellicle functions like a flexible coat of armor, preventing the protist from being torn or pierced without compromising its range of motion. The majority of protists are motile, but different types of protists have evolved varied modes of movement. Some protists have one or more flagella which they rotate or whip. Others are covered in rows of tufts of tiny cilia that they beat in coordination to swim. Still others send out lobe-like pseudopodia from anywhere on the cell, anchor the pseudopodium to a substrate, and pull the rest of the cell toward the anchor point. Some protists can move toward light by coupling their locomotion strategy with a light-sensing organ. How protists obtain energy. Protists exhibit many forms of nutrition and may be aerobic or anaerobic. Photosynthetic protists, photoautotrophs, are characterized by the presence of chloroplasts. Other protists are heterotrophs and consume organic materials, such as other organisms, to obtain nutrition. Amoebas and some other heterotrophic protist species ingest particles by a process called phagocytosis, in which the cell membrane engulfs a food particle and brings it inward, pitching off an intracellular membranous sac or vesicle called a food vacuole. This vesicle then fuses with a lysosome, and the food particle is broken down into small molecules that can diffuse into the cytoplasm and be used in cellular metabolism. Undigested remains ultimately are expelled from the cell through exocytosis. Figure 13.4 shows the stages of phagocytosis. Some heterotrophs absorb nutrients from dead organisms or their organic wastes, and others are able to use photosynthesis or feed on organic matter, depending on conditions. Reproduction. Protists reproduce by a variety of mechanisms. Most are capable of some form of asexual reproduction, such as binary fission to produce two daughter cells, or multiple fission to divide simultaneously into many daughter cells. Others produce tiny buds that go on to divide and grow to the size of the parental protist. Sexual reproduction, involving meiosis and fertilization, is common among protists, and many protist species can switch from asexual to sexual reproduction when necessary. Sexual reproduction is often associated with periods when nutrients are depleted or environmental changes occur. Sexual reproduction may allow the protist to recombine genes and produce new variations of progeny that may be better suited to surviving in the new environment. However, sexual reproduction is also often associated with cysts that are a protective resting stage. Depending on their habitat, the cysts may be particularly resistant to temperature extremes, desiccation, or low pH. This strategy also allows certain protists to wait out stressors until their environment becomes more favorable for survival or until they are carried, such as by wind, water, or transport on a larger organism, to a different environment because cysts exhibit virtually no cellular metabolism. Protist diversity. With the advent of DNA sequencing, the relationships among protist groups and between protist groups and other eukaryotes are beginning to become clearer. Many relationships that were based on morphological similarities are being replaced by new relationships based on genetic similarities. Protists that exhibit similar morphological features may have evolved analogous structures because of similar selective pressures, rather than because of recent common ancestry. This phenomenon is called convergent evolution. It is one reason why protist classification is so challenging. The emerging classification scheme groups the entire domain Eukaryota into 6 supergroups that contain all of the protists as well as animals, plants, and fungi. These include the Excavata, Chromalveolata, Rhizaria, Archaeplastida, Amoebozoa, and Opisthokonta. The supergroups are believed to be monophyletic. All organisms within each supergroup are believed to have evolved from a single common ancestor, and thus all members are most closely related to each other than to organisms outside that group. There is still evidence lacking for the monophyly of some groups. Human pathogens. Many protists are pathogenic parasites that must infect other organisms to survive and propagate. Protist parasites include the causative agents of malaria, African sleeping sickness, and waterborne gastroenteritis in humans. Other protist pathogens prey on plants, affecting massive destruction of food crops. Plasmodium species. Members of the genus Plasmodium must infect a mosquito and a vertebrate to complete their life cycle. In vertebrates, the parasite develops in liver cells and goes on to infect red blood cells, bursting from and destroying the blood cells with each asexual replication cycle. Of the 4 Plasmodium species known to infect humans, P. falciparum accounts for 50% of all malaria cases and is the primary cause of disease-related fatalities in tropical regions of the world. In 2010, it was estimated that malaria caused between 0.5 and 1 million deaths, mostly in African children. During the course of malaria, P. falciparum can infect and destroy more than one-half of a cell's circulating blood cells, leading to severe anemia. In response to waste products released as the parasites burst from infected blood cells, the host immune system mounts a massive inflammatory response with delirium-inducing fever episodes as the parasites destroy red blood cells, spilling parasite waste into the bloodstream. P. falciparum is transmitted to humans by the African malaria mosquito, Anopheles gambiae. Techniques to kill, sterilize, or avoid exposure to this highly aggressive mosquito species are crucial to malaria control. Figure 13.6, it's a light micrograph, 100x magnification of red blood cells infected with P. falciparum, and those are shown as purple. Trypanosomes. T. brucei, the parasite that is responsible for African sleeping sickness, confounds the human immune system by changing its thick layer of surface glycoproteins with each infectious cycle. The glycoproteins are identified by the immune system as foreign matter, and a specific antibody defense is mounted against the parasite. However, T. brucei has thousands of possible antigens, and with each subsequent generation, the protist switches to a glycoprotein coding with a different molecular structure. In this way, T. brucei is capable of replicating continuously without the immune system ever succeeding in clearing the parasite. Without treatment, African sleeping sickness leads invariably to death because of damage it does to the nervous system. During epidemic periods, mortality from the disease can be high. Greater surveillance and control measures have led to a reduction in reported cases. Some of the lowest numbers reported in 50 years, fewer than 10,000 cases in all of sub-Saharan Africa, have happened since 2009. In Latin America, another species in the genus, T. cruzi, is responsible for Chagas disease. T. cruzi infections are mainly caused by a blood-sucking bug. The parasite inhabits heart and digestive system tissues in the chronic phase of infection, leading to malnutrition and heart failure caused by abnormal heart rhythms. An estimated 10 million people are infected with Chagas disease, which caused 10,000 deaths in 2008. Figure 13.17. Trypanosomes are shown in this light micrograph among red blood cells. The trypanosomes are in purple. The red blood cells are the light pink. Plant parasites. Protist parasites of terrestrial plants include agents that destroy food crops. The oomycete Plasmopara viticola parasitizes grape plants, causing a disease called downy mildew. Grape plants infected with P. viticola appear stunted and have discolored, withered leaves. The spread of downy mildew caused the near collapse of the French wine industry in the 19th century. Figure 13.18 shows the downy and powdery mildew on a grape leaf caused by infection with P. viticola. And then on the right, B, this potato exhibits the results of an infection with P. infestans, the potato late blight. Phytophthora infestans is an oomycete responsible for potato late blight, which causes potato stalks and stems to decay into black slime. Widespread potato blight caused by P. infestans precipitated the well-known Irish potato famine in the 19th century that claimed the lives of approximately 1 million people and led to the emigration from Ireland of at least a million more. Late blight continues to plague potato crops in certain parts of the United States and Russia, wiping out as much as 70% of crops when no pesticides are applied. Beneficial protists. Protists play critically important ecological roles as producers, particularly in the world's oceans. They are equally important on the other end of food webs as decomposers. Protists as food sources. Protists are essential sources of nutrition for many other organisms. In some cases, as in plankton, protists are consumed directly. Alternatively, photosynthetic protists serve as producers of nutrition for other organisms by carbon fixation. For instance, photosynthetic dinoflagellates, called zooxanthellae, pass on most of their energy to the coral polyps that house them. In this mutually beneficial relationship, the polyps provide a protective environment and nutrients for the zooxanthellae. The polyps secrete the calcium carbonate that builds coral reefs. Without dinoflagellate symbionts, corals lose algal pigments in a process called coral bleaching, and they eventually die. This explains why reef-building corals do not reside in waters deeper than 20 meters. Not enough light reaches those depths for dinoflagellates to photosynthesize. Figure 13.19. Coral polyps obtain nutrition through a symbiotic relationship with dinoflagellates. Protists themselves and their products of photosynthesis are essential, directly or indirectly, to the survival of organisms ranging from bacteria to mammals. As primary producers, protists feed a large proportion of the world's aquatic species. On land, terrestrial plants serve as primary producers. In fact, approximately 1/4 of the world's photosynthesis is conducted by protists, particularly dinoflagellates, diatoms, and multicellular algae. Protists do not create food sources only for sea-dwelling organisms. For instance, certain anaerobic species exist in the digestive tracts of termites and wood-eating cockroaches, where they contribute to digesting cellulose ingested by these insects as they bore through wood. The actual enzyme used to digest the cellulose is actually produced by bacteria living within the protist cells. The termite provides the food source to the protist and its bacteria, and the protist and bacteria provide nutrients to the termite by breaking down the cellulose. Agents of decomposition. Many fungus-like protists are saprobes, organisms that feed on dead organisms or the waste matter produced by organisms. Saprophyte is an equivalent term, and are specialized to absorb nutrients from non-living organic matter. For instance, many types of Oomycetes grow on dead animals or algae. Saprobic protists have the essential function of returning inorganic nutrients to the soil and water. This process allows for new plant growth, which in turn generates sustenance for other organisms along the food chain. Indeed, without saprobic species, such as protists, fungi, and bacteria, life would cease to exist as all organic carbon became tied up in dead organisms.